Hi guys, I was just going to chat today about chapter 5 where we talk about membrane, structure, and function. So with the plasma membrane, we know that the plasma membrane is super important because it helps a cell to create compartments and also allows for inside separation as well. And so by separating everything, it allows all the organelles to kind of do exactly what they're supposed to be doing, but individually and separately as separate reactions. So if you recall, when we talked about chapter 3, we mentioned phospholipids because we were talking about lipids, and then also we talked about steroids and proteins as well. So all of those are big major components of the plasma membrane. The plasma membrane also has what are called glycoproteins and glycolipids, and so when you see this, you see carbohydrate chains that are attached to, and remember carbohydrates are like sugars that are actually attached to proteins and lipids. And so with that, you actually get to have also additional things associated with that plasma membrane, and that's kind of why it's referred to as a fluid mosaic model. So it has the consistency of oil, but then also it has all these different components like cholesterol to prevent it from being too ooey gooey, and those proteins they play a whole ton of important roles, which we will talk about. So there can be proteins that are extended through the membrane. Those are called integral proteins, or there can be peripheral proteins where you'll have it on either side. There are certain proteins, and if I were you and if I were studying for this next exam, I might know 3 of the 6 commonly found proteins associated with that plasma membrane. So one of those are channel proteins. So with channel proteins, there's just a channel that works through the plasma membrane and allows things to pass through. And these things are generally larger and like polar things or things that can't just freely pass through that phospholipid. And so it's kind of selective. Carrier proteins are those kind of important proteins because they essentially receive an ion or a molecule, and they change shape to let that molecule pass. And a really good example of that is the sodium-potassium pump in nerve cells. So they really do change shape to allow sodium to go one way and potassium to go the other way. Cell recognition proteins are super important. These guys actually help the immune system recognize you in comparison to an unknown pathogen. And so then if some unknown pathogen were to come in, then your body would launch this big, huge immune response and essentially help you fight off that unknown defender, which is great unless that unknown defender is actually a known defender and something that you want to be there, like your new kidney or something that you needed. So sometimes your body is so good at cell recognition proteins that it tries to fight off things that you really need, like if you were to get an organ transplant. Receptor proteins are shaped to allow specific molecules to bind to it, and that binding causes some kind of change in the cell. And often it's associated with like a signal transduction pathway. Those signal transduction pathways initiate cellular response. Enzymatic proteins are those proteins that essentially help catalyze chemical reactions in a cell. So they're very important. And by catalyze, they kind of help enzymes. They do help things process better, if you will. So like if you're looking at like breaking down things, like lactase has the role of degrading lactose. And so if you lack that lactase in your body, then what you'll find is that you're not able to process lactose. And when you can't process lactose, your tummy lets you know right away. Junction proteins are those proteins that just form like junctions between cells. And so the really tight junctions act to help cells stick together. Some connect plasma membrane to the cytoskeleton. And there is peptidoglycan between plant cell wall junctions as well. So that's a structural carbohydrate, if you remember. Now, when things cross the plasma membrane, it's generally regulated by that plasma membrane. So it's selectively permeable. It doesn't just let everything cross willy-nilly. It's selective. And so some of that movement is passive. And by passive movement across that membrane, what you'll find is that there's no energy requirement. Certain non-charged molecules move freely down a concentration gradient. So it'll move from areas of high concentration to areas of low concentration. One of the ways that we look at things moving in and out of a cell is by having an understanding of the concept of diffusion. And so with diffusion, you'll see molecules move from areas of high concentration to areas of low concentration until there's an equilibrium reached. And so, and we use diffusion. We're familiar with diffusion because we're required to, well, maybe not required, but we appreciate diffusion. Like when we want to smell really nice, we'll spray high concentration of perfume or cologne on our bodies, and then we hope that it goes from that area of high concentration to lower concentration and diffuse out to others. Or like if we buy an air freshener for our car, our hope is that that high concentration of air freshener scent will diffuse from that air freshener all the way out to our vehicle to make our vehicle smell really nice. We're also dependent on diffusion for gas exchange in our lungs. And so obviously when high concentrations of oxygen build up in the alveoli, they're going to want to go from that high concentration to the lower concentration in the capillaries. And so that's how oxygen gets from our lungs out. Osmosis is a special type of diffusion, and so you're looking, instead of just any particle diffusing or moving through the air, with osmosis, you're looking at the movement of water specifically through a selectively permeable membrane. It still works to go from areas of high concentration to low concentration, but you're looking at water moving across the membrane, not just particles moving wherever. Water across the membrane. And so because you're going from high concentration on one side of a membrane to low concentration on the other side of a membrane, that's where you'll see a buildup of osmotic pressure. And water doesn't just go directly across that membrane. It actually crosses the membrane via these special channels called aquaporins. And so they help speed up the transfer of water. And it's a relatively newer protein that was discovered, like scientifically on that scale of discovery, it's relatively newer. When we're looking at like osmosis and the movement of water from one side of a membrane to the other, we consider different types of solutions based on that water movement. Okay, and we'll see these types of solutions in lab as well. So when you have an isotonic solution, what happens is you have the same amount of particles or solutes as you do water, and that's the ideal situation because there's no movement to one side or the other. It's just equal. There's an equilibrium. With a hypotonic solution, what you'll see is more water outside of the cell than inside. And so that means the solute particles in the solution are lower than in the cell. And so when you see that hypotonic situation, water is going to go from higher concentration outside of the cell and flow into the cell, which is fine if you're a plant cell and you have a cell wall, but if you're an animal cell and you have only a plasma membrane, that could actually cause your cells to rupture or lyse, if you will. A hypertonic solution, you'll see more water inside the cell than outside of the cell, and there's a higher solute concentration in the solution than in the cell. And so when that happens, water is going to leave that area inside the cell where there's higher concentration, and it's going to want to go to where it's lower concentration. And this shows you some examples of blood cell in these different types of solutions and how they behave. This is a really good image that shows you osmosis in animal and plant cells. And so you can see in both an isotonic solution, everything's in equilibrium. And then in a hypotonic solution, water is going to enter the cell, and that could cause an animal cell to rupture. But a plant cell is just fine. It has that giant vacuole that can store all of that. And then a hypertonic solution, water is going to actually leave the cell, and that can cause crenation in animal cells and plasmolysis in plant cells. Obviously, some organisms find themselves in a position where they have to be adapted to living in water. And if you think about that, fish are just a bunch of cells that live in water, and they have to live and to adapt to either freshwater or saltwater. And so that makes a difference in how they handle those situations. So like saltwater fish live in a solution that's hypertonic to their cells. And so what they do to adapt is they drink a lot of water and they excrete little to no waste, so that way they don't lose water to their environment. Freshwater fish live in a hypotonic solution, and so to avoid water just flowing in and causing all of their cells to rupture, they don't drink water because they're already living in fresh water. And they urinate a lot, and that kind of accounts for the high ammonia in your home aquarium. Freshwater fish have to pee a lot to get rid of the water so their cells don't rupture. Now, passive movement across membranes require no energy, that's what we've been talking about, and very little effort. Mind you, the rate of diffusion is really based on the size, the shape, the electrical charge, even the temperature in the environment. Facilitated diffusion is still passive diffusion. You're still seeing things move from areas of high concentration to low concentration. So another kind of transport is active transport. Active transport requires an energy input. And so what happens is materials are moved against their concentration gradient. So you're moving from areas of low concentration to high concentration. And when that happens, energy is required. The best example that I can think of for active transport is a sodium-potassium pump. So that's a membrane, a carrier protein, and it basically maintains the potassium concentration inside your cell. And the protein will change shape based on the interaction between the ATP molecule and breaking that down, and then that will allow sodium and potassium to move in and out of the cell. So this is a really great image of the sodium potassium pump. And so if you look, you also have this in your textbook. So ATP attaches to that protein carrier. The energy between that second and third phosphate is super high energy and kind of wily. And so breaking that off of that third phosphate will, to attach it to that carrier, changes the shape to allow then the sodium to be loaded up into the carrier, and then the carrier will change shape and allow sodium to be released, and then potassium is taken in, and then the release of that phosphate from that carrier changes the shape of the carrier to release potassium inside. And the whole purpose of this is to trigger a nerve impulse that would elicit some response. This is a funny meme, I love it. Sometimes things have to go in and out of the cell that are a little bit big, and so bulk transport imports and exports larger materials. So essentially, endocytosis and exocytosis is things. Endocytosis is entering the cell, and exocytosis is exiting the cell. And so with endocytosis, examples of that are phagocytosis, pinocytosis, and receptor-mediated endocytosis. With exocytosis, essentially things are just packaged up into a vesicle, and that vesicle fuses with the plasma membrane and releases the product. Endocytosis is basically like divided by the things that it's getting. So phagocytosis is often called cell eating because that's where large solid material is taken in and then enveloped in a vesicle. Pinocytosis is referred to as cell drinking, if you will, and that forms vesicles around small material or liquid. And then receptor-mediated endocytosis is basically binding at a specific site in the cell membrane, and that moves materials in and out of the cell. This is just an image of 3 forms of endocytosis. Please look at those. Exocytosis basically happens as vesicles from the Golgi apparatus contain materials that fuse with the cell membrane. So those might be hormones, neurotransmitters, digestive enzymes, whatever. So another important component of a cell is the cell surface. And so the cell surface, or the area that's around the cell that's actually secreted by that cell, is referred to as the extracellular matrix. With the extracellular matrix, it's made up of collagen, elastin, integrin, all those things, fibronectin. These are things that help resist stretching, provide resilience. There's adhesive proteins, so cells can join together. And then, of course, integrin is important in cell signaling. Cells don't send text messages, they send chemical messages. So cells do join together. You can see adhesion junctions where you'll see just like different, essentially adhesion junctions. You'll see desmosomes where you'll have kind of a little spot of attachment between cells, or you'll see these tight junctions that form impenetrable barriers, and those barriers that you cannot cross actually help to keep in some substances that we wouldn't want to get out. Like there are tight junctions with our intestines, so we don't have, toxic things released into our bodies. And then there are gap junctions, and this is where you'll have plasma membrane channels that get joined, and then you'll see more communication between those cells. And that's super important for cardiac muscle to communicate with the other cells. For plants, plant cells actually have plant cell walls, which is super made of cellulose, and it's permeable. And then there are also these plasmodesmata that help things pass between those cells.